Okay, guys, so let's start today's lecture. Ooh, that's hot. Okay, so let's start today's lecture. I'm going to announce you guys the heroes of molecular biology 1 because you're here. It looks like less than 15% of the class is really serious about taking this. And it also gives me an opportunity. You chose to put your bodies in this room, right? And let's also put our minds in this room because I'm going to remind you guys that you will get better at what you pay attention to, right? And in the first class, I said there's a lot of neuroscience and psychology studies suggesting that human brains cannot multitask cognitive functions. And because of this, having laptop in the classroom is going to hinder your learning. And there's a lot of research backs up this. And not only your learning, it will also hinder the people around you, their learning, right? So if you look at, for example, here, the people who are sitting, they're not multitasking, but they see someone multitasking, they will do considerably worse in a test that they take afterwards, okay? So in addition to shutting off your laptops, the other thing that I reminded you is that taking notes by hand is a lot better taking notes by laptops because when you take notes by laptops, you tend to dictate, okay? Then that's not a very effective way of taking notes. Actually, if you summarize what's going on and use your own words, paraphrase, you're going to learn much, much better. And here's some data about that. So, for example, this is, again, test results in the y-axis. For factual questions, like basically you learn and then you just spit out the fact, the difference between people who use laptops to take notes versus pen to take notes is not that different, right? But for conceptual questions, the difference is much, much greater, right? So people who paraphrase, who take notes with their pen, do a lot better in understanding and answering conceptual questions. And you've seen in the first midterm that we tend to include a lot of conceptual questions in the test. So it's going to help you. So basically what you can do maybe to improve your grades in midterm and the final will be turn off your devices, put them away. Again, this is your decision. I'm not dictating you, right, to do this. But I'm telling you that it's going to help if you do this. And you can also start taking notes by hand. It's also going to help. And I promise you, if your ex puts a picture out for drinks, it's going to be there in 75 minutes. Okay, you don't have to see this now. All right. Okay, so let's start the lecture. So today we're going to talk about transcription regulation, and there's going to be a lot of techniques, and a lot of these techniques are going to be conceptual, so pay attention. Why do we spend so much time talking about transcription regulation? Because to understand biology of humans and animals, we need to understand why a single genome can create the cells that do different functions, and they do have different structures, right? So in our bodies, if you look at a nerve cell, it's going to look something like this, where this is the cell body, and it's going to have all these extensions, and that's going to look very different from a muscle cell. And the reason why these two things are looking very different is because they contain protein and RNA products that are different from each other, right? So the nerve cells will contain this protein acetylcholinesterase is going to make the acetylcholine, and the muscle cells is going to contain the receptor so that it can actually take the signal from the nerve cells, right? So they're going to basically, different types of cells are going to express different genes, making specific proteins for specific cell structure and function. So this is what we think about when the genome is used differently in different cells. And the main reason this occurs is that different cells have different transcription factors that define a cell's identity. So in muscle cells, these muscle cells have a transcription factor called MyoD, it binds to DNA in a sequence-specific manner. So it recognizes certain sequences in the DNA, short sequences, and these sequences are found in the gene regulatory regions of genes that need to be expressed in the muscles, right? So this MyoD is going to bind to a DNA sequence that we call the E-box, and these E-boxes are found at the promoters of and enhancers of genes that are required for muscle cell structure and function, such as myosin light chain, heavy chain gene encoding for the creatine kinase, acetylcholine receptor, etc. So you will then what is happening here is that this transcription factor MyoT will bind and coordinately regulate. That means it regulates many genes that are going to be expressed in the muscle cells. Okay? So that's the basis of gene regulation. So transcription factors are the window into our understanding of how the genome sequence is interpreted, because they can bind to specific DNA sequences and turn genes on or off. Okay? So when you look at the genome, yeah?
Did you say that they bind to promoters or enhancers?
They bind to both promoters and enhancers. Transcription factors do bind to gene regulatory regions, and in eukaryotes, these include both promoters and enhancers, right? Bless you. Okay, so what you have here is that you have the genome, right? And then what if you looked at where these transcription factors bind, so you will find transcription factor 1 binding to the promoter of this gene, 2 binding to promoter of this gene, 2 and 3 bind to here, etc., right? So, and this whole binding and the regulation of all these set of genes is going to determine the transcriptional program for a cell type. So how do these binding regulated? For example, in cells, signals such as hormones or other transcription factors will control a transcription factor's expression or binding, and then a set of transcription factors will bind to their target sequences in the DNA at enhancers and promoters, and the combinations of transcription factors in a cell will determine the transcriptional program of that cell. Okay? So this is a simplified picture of a small genome with 3 genes, right? And there are 5 transcription factors that are regulating them. In reality, the picture is more complex. For example, in the single-cell eukaryote yeast, you have about 300 sequence-specific transcription factors. In worms, we have about 1,000 of them. And in humans, the estimated number of sequence-specific transcription factors is 2,000. Okay? So there are 2,000 genes that are interpreting the genome by going to their specific sites and turning the genes on or off. All right. So if we really want to understand how the genome is interpreted, how transcription factors work, we need to answer several questions, right? And the first question is, we need to know where the transcription factors bind. So how do we do this? I'm going to go through some techniques, okay? So the first thing that we're going to talk about is deletion analysis and reporter assays, and these assays are geared towards answering questions such as where are the gene regulatory elements in the genome? Where do the transcription factors bind? All right, so reporter assays, you're going to see this over and over again, okay? So the idea is that what you want to have is you want to have the expression of a reporter protein that is biologically inert for the cell that you're analyzing. It's not produced by the organism under study, so that its production doesn't interfere with the normal cellular functions. And the products are easy to assay or visualize, right? And a very good example of this would be GFP, which is a fluorescent protein that is not going to interfere with cell function. So the idea is this. You suspect a small DNA fragment that can activate transcription of a certain gene, okay? So what you do is you clone that small DNA fragment upstream of a reporter gene, right? And then if this DNA fragment is acting to activate the transcription of this gene, such as GFP, this gene will be transcribed, it's going to be translated, and it's going to create a product that you can see, right? So if you look at your cells, that is containing this construct, if they are appearing green, you know that the transcription of the reporter has happened. And if they don't look green, you know that it didn't happen. Okay? So it's reporting whether the transcription occurred or not. So that's a reporter. All right. So what are other examples of reporters that you've heard about already from Professor Siegel? Any examples? GFB. I think you also heard about LAC-Z, right? Beta-galactosidase. It turns X-gal blue when it's present in the cells. You can also, so these are transcriptional and translational reporters because they are GFP and LAC-Z. You're measuring the amount of protein, right? So it needs to be transcribed and translated. You can also use simple transcriptional reporters by using in situ hybridization, right? So then you would be measuring the amount of the RNA rather than amount of protein, right? So there are different kinds of reporters that you can use. How do you use reporters to find regulatory regions? Okay, here's one example of how you can do this. So what we're looking at is we're looking at here a gene. We know its transcription start site, and we know its upstream region of the TTR RNA, or TTR DNA, this gene's upstream region. And we want to know which part of this DNA is actually required for the activation of this gene's transcription, right? So what you do is you use molecular cloning techniques to produce fragments of different length that contain the transcription start site and little pieces of this upstream region of the gene. What kind of technique can you use to make these shorter and shorter products? What would you do? You learned cloning. What technique can you use to produce these fragments? Come on, guys. It's so famous that it was in POB. It was probably in your high school. I probably told it to my 8-year-old daughter. It's that famous. PCR, right? So what you can do is you can make primers, right? So they are going to, if you make a primer like that and amplify this region, you're going to make a large product, right? If you make a primer that's downstream, and that's going to amplify and make that product, etc., right? And you learned in molecular cloning that you can make these PCR products with restriction enzymes on each end so that you can clone these into a plasmid vector, right? Clone them into a plasmid vector, right? All right, so what is happening here is that you take your plasmid vector and it contains your reporter gene, right? And then you insert your fragments right here, right upstream of the reporter gene, right? It's going to have different lengths of these fragments. And what it's going to happen is you're going to have a set of plasmids that contain the regulatory elements of different sizes upstream of your reporter gene, right? And so we call these deletion analysis because as you make it shorter and shorter, you're deleting more and more of the upstream sequences. Does that make sense? So far, so good. Okay, so what do you do next? What you do is you take all of these plasmids and you transfect your cell with each of these plasmids, okay? So you take each of these plasmids, you transfect your cells, and then look for the reporter mRNA or reporter enzyme. So if this, in this case, if this was a GFP, you would look for GFP, how green the cells are, right? And you're going to have a measure of whether that enzyme is expressed, present or not, right? So you're going to measure that and you're going to express that as a number or as some plus signs, right? So this would be that reporter gene is expressed. This is expressed at a lower level, right? So you have multiple pluses for high expression, so the cells are really green. You will put 1 plus for lower expression, the cells are a little less green, right? And then you would put a minus for no expression, right? So you're measuring the reporter gene expression. And you're doing this for each of the plasmids that you generated. Okay, so how do you then use the data? So what you do is you align your fragments that you cloned right next to the reporter gene expression, and then look, did a deletion of a certain region in my construct caused a reduction in expression, right? So here, this deletion of this region, did it cause a reduction in expression? No, right? So you still have high expression equal to the longer fragment. How about deletion of this region? Did it cause a reduction? Yes, right? When you have this construct 3 compared to construct 2, which is shorter, it's shorter by this fragment, you now have a reduction in reporter activity. You went from lots of expression to little expression, right? And how about the deletion of this region? So between construct 3 and 4, is there any difference in activity? No. So that region is not responsible for activating. How about deletion of this? Yes, right? So you grow from plus to a minus, right? So something in this region is important, right? So essentially by doing that deletion analysis, you can decide which regions of the fragment that I cloned is important for activating that gene. So which are the gene regulatory elements responsible for activating my gene? So far, so good? All right. So people can do this in one gene at a time, okay? And a single gene in mammals can have multiple regulatory regions. So it will have a promoter, and it can have many different enhancers. So this is an example from your book. So this is the PAX6 gene. This is a transcription factor, and its expression is controlled by these enhancers shown in green, blue, etc. Right? And what is going to strike you is that the enhancers can work in a modular way. What I mean by that is that you can have a segment of the DNA near the gene, right, that will promote the expression of this gene only in pancreas, and another region and another enhancer will express it in retina, and another will express it in certain cell types, etc. So a single gene will have multiple enhancers, and each of these enhancers will be responsible for activating that gene in a given cell type. Okay? So it can drive expression in different genes. How do you actually find these enhancers? What did the researchers do to determine that this little piece of DNA in the intron of the gene is capable of activating the gene? So the way that they did that is they took big fragments around the gene and they cloned it to the upstream of a basal promoter. A basal promoter is basically the DNA segment that contains the consensus sequences that are bound by general transcription initiation factors to activate a gene. Okay? So what you have here is that this basal promoter is going to recruit all the general transcription factors. Give me the name of a general transcription factor. In transcription initiation.
D.
TF2D. What else? 2A, B, C, D, E. What's the most important? That comes first. Very good. TBP, target binding protein, right? So it would have sequences there that will be recognized by these general transcription factors. binding of simply just the general transcription factors is not enough to turn on the gene high enough, okay? It's not going to turn on gene expression very much. That's what transcription factor like activators do, is they increase the amount of transcription, right? And so these sequence-specific transcription factors are going to be binding to these elements such as enhancers, and they're going to activate the transcription of this reporter, which in this case is LAC-Z, that you can use to look by using the assay to visualize the LAC-Z expression, right? So they're putting the fragments to be tested upstream of a basal promoter, and then they're seeing if that promotes expression of LAC-Z, and then they're also asking in which tissue is this fragment that I'm testing is increasing expression, right? So in this example, the fragment that is that has been tested is expressing the gene, for example, in the eye, right? So they can look at the whole mouse embryos and then look at where this thing is expressed. So you can then determine which fragments are capable of activating transcription and which fragments are not capable of activating transcription, and make this kind of a figure showing, okay, these are the fragments that we know increase the transcription. When you go like this, you want to minimize the work that you have to do, right? So if you have a gene that's 100 kb, right, you can't take 100 base pair pieces and look each and every single one of them if they're capable of activating transcription. That will take years, and that's not very a good, smart way to do this, right? So what we usually do is we usually take big chunks at first, right, and then see if they can activate. Now, if you have a big chunk of DNA that you know is capable of activating, how can you find the smaller chunk that's important? What can you do? So you have a big fragment, you have a 10 kb fragment, but you want to know exactly which segment within this is important for activating that gene.
Is this the thing where you make it so that the protein's bound really tightly and then cut up everything else, but it won't cut up that because there's protein on it?
You could do that, but this is too big of a fragment to do that.
You can try deleting small segments.
Very good. Deletion analysis. We just learned it 2 slides ago, right? So what you can do is you can make smaller and smaller fragments, and to figure out which regions of that larger fragment is important for activating the transcription, and then so you can narrow down the segment of the genome that's responsible for activating that transcription of that gene in the retina, right? So you can find that out. All right. So unlike promoters, enhancers are orientation independent. This is same in prokaryotes and eukaryotes. So in a lot of the enhancer assays, we simply put the fragment to be tested upstream of the basal promoter, right? But people have done experiments to test the directionality, to see if enhancers are directional, and they use these reporter experiments to do this. So to test if a piece of fragment that you've used, if it's directional or not directional, what experiment can you do? So you have this set up, right? And you want to know, is this fragment capable of activating this LAC-Z independent of its direction or independent of its localization, right? Because you can find enhancers in introns, upstream of genes, downstream of genes, anywhere. You can reverse, right? You can reverse this fragment. If it still works exactly the same, then it's an enhancer, it's not a promoter. What else can you do?
You could put it after.
You can put this fragment downstream of the reporter and it will still work. So there are a lot of enhancer assays where you would clone the fragment that you're testing in the 3' downstream of the gene so that you can make sure this is an enhancer and not a promoter, right? Okay, so that was determining what fragments in the genome are controlling a gene's expression or not, right? And then what the next thing that you want to know is which DNA sequences within these elements is important, right? And there are several assays in which you can do this. One of them is called the linker scanning mutation assay. It's a fancy name, but a very simple idea. So what you do is you have a region that you determine controls the expression of a gene. You clone it upstream of your favorite reporter gene, and then you have an assay to measure the expression of this reporter gene. Okay? So that's pretty much it so far, same thing. And then what you do is you make versions of this clone, right? So you make versions of this control region in which you randomly mutated certain parts of the control region. So these mutations here are shown as these dashed lines. So you jumbled the sequences over there using PCR or some other kind of method, and you change the sequence of this region. And you do this in in different regions, stepping all across this control region, okay? And then you ask which of these segments, the mutation of which of these segments actually affected the reporter expression, right? So let's go here. So did the mutation of this segment affect the reporter expression? No. So I can conclude that these sequences here are probably not important for this fragment to function, right? Those sequences are probably not the sequences where my transcription factors bind. What about mutation of this region? Did it affect expression of the reporter? Yes, right? So I say, okay, I know it's kind of like a puzzle, right? I know that this region was not important, right? And that's overlapping with this. I can say this region is not important at all. But in this region, there might be sequences that are important because when you mutate them, you affect the activity of this reporter, right? And then you go down and do the same thing for each and every single one of them. It's basically like a puzzle, right? So in between here and here, there's a big drop in the activity, right? So you know the sequences here are not important, so you can line them up with the next construct and say, okay, these are sequences not important, but these sequences contain something that's important because you no longer have reporter activity. But in your next construct, you actually mutated these regions and it made no difference in the reporter activity. So you go back and say, I know that this little piece here is also not important, right? And then you can say, this little guy here contains sequences that are important for this control element's activity. So far, so good?
What did you say cause the mutation? What do you use to cause the mutations?
There are different techniques that you can use. I don't do this assay very much, so I don't know all the details about it, but there are ways that you can introduce random mutations in a piece of DNA in a plasmid. Okay, so we can do a Google search and find out what people use. All right. There are actually even kits to do this, so that's part of the reason why I don't know, because you just use the kit. All right. So another method to find out where your transcription factor binds within. So the linker scanning mutation assay doesn't tell you where your transcription factor binds. What it tells you is that there are sequences here. These sequences are important for this control element's function. It doesn't tell you that your transcription factor is binding to that sequence, right? So how do you test that? You have a transcription factor. You know that if you mutated, you did a mouse knockout, you no longer have eye development, right? And you want to know what genes is this transcription factor controlling, what sequence does it bind to, and where does it bind, right? So you need to know these things, and what do you do? One experiment you can do is called DNase footprinting assay. All right, so here's an assay that you really need to pay attention to. It's a little bit tricky, okay? So the principle of DNA's footprinting assay is that if you have a protein that binds to DNA stable enough, okay, it's going to protect that piece of DNA from cutting by the enzymes that can cut DNA, okay? So if you have a protein that is binding to a piece of DNA, it's going to protect the cutting of that DNA by an enzyme which is called DNase1. It cuts DNA, and that's why it's named DNase. Okay, so if your protein is sitting at a site and then you cut with DNase, it's going to cut all over here and all over here and just basically digest this DNA out, and this little piece here, that little DNA piece here, is going to be protected. And that's what we call the footprint. It's the footprint of the protein that's protecting it. Okay? So far, so good? All right. So how does this assay work? You take a DNA fragment that you know has sequences that are important for its function, and you suspect that your transcription factor binds to this fragment. You want to know where does your transcription factor bind. First, you take naked DNA, so no protein in it. You take that DNA fragment, and what you do is you label one strand using a radioactive incorporation of radioactive ATP to that 5' end of the DNA, right? So the radioactivity is going to label just one strand of that double-stranded DNA. And now what you do is you take that DNA, put it into a tube, and then throw in some DNase, right? And the DNase is going to cut randomly across this fragment. And you adjust the DNase concentration in that it's not going to completely chew the fragment. It's going to be just enough to introduce single cuts into the DNA. Okay, so what it's going to produce is it's going to produce all of these little cuts. The DNase is an interesting enzyme. It creates nicks. Instead of cutting double-stranded, it makes single-stranded cuts, right? So the product of, for example, this cut, if you denature the DNA, is going to be a little fragment that contains the label, right? And it's going to have the other fragment that doesn't contain the label here, the other strand that doesn't contain the label. And then you'll have the full strand on the other side, the complementary strand, that's going to be full length because it's not cut, right? So the product of this cut here, if you denature the DNA, is going to be 3 strands, one little, one big, and then one large. Yeah?
How does it know which strand to cut?
It doesn't. It's random.
Then how does it protect the non-labeled strand?
It doesn't protect the non-labeled strand. It cuts that non-labeled strand. In this picture, they have somewhat simplified it. And it's also going to cut here, it's going to cut here, it's going to cut there, it's going to cut there, etc., etc. It's going to cut on both strands, okay? And then in a second tube, your experimental tube, you're going to have your protein bind to DNA first, and then you're going to incubate it with the DNase. And in that tube, what's going to happen is that the DNase is going to cut again at regions. It's going to cut here, it's going to cut there, it's going to cut here, here, etc. But it's not going to be able to cut where the protein is sitting. Okay? So the product of this, for example, this cut here would be, let me see here. The product of this cut here is going to be a fragment that is labeled on one end, another fragment here, DNA strand there, and then a long strand here. Okay? So then you analyze your data by denaturing all the DNA and running them on a gel. Let me see here. Okay. All right. And the gel is going to look something like this. Okay? So what you have here is that you have mixed your TBP with your DNA. So you're looking at title binding protein and the promoter sequence of some gene that you have isolated. If there is no protein, what is going to happen is you're going to have all kinds of different fragments that are generated by this random cut of the DNA, right? So the DNA is going to cut here, 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 and what you're seeing is that you're seeing only the labeled products. These gels, you use a radioactive detection system so that you only see the strands that contain the radioactive label, right? So you're going to have all kinds of sizes of DNA. Does that make sense? So far?
So it doesn't even account for the second DNA strand?
You're not going to see this other guy here. Why can't you see that other guy? Because it's not labeled and you're detecting the radioactivity in the gel. Okay? So that's why. Yep.
Is there a chance that with the longer fragments that it would cut multiple times and you would get more smaller fragments?
That's right. And that's why you have to actually titrate your enzyme so that it doesn't happen. Because if all your fragments are down here in the bottom of the gel, your assay is not going to work, right? So you're going to make sure you have just the right amount of enzyme to see this. All right, so what happens in the tube when you have the protein, what will happen is that you're going to produce the fragments, right, that are random like this, right? But you're not going to produce any fragment that's long into and goes into the DNA, the binding side of this protein, right? So you're not going to have those fragments that are going into the binding side of the protein. And when you run the gel, what you're going to see is that those fragments are not going to be produced because the DNAs can't cut, right? So you're going to see a gap. You're not going to see those things where your protein binds. And you run also a marker here to tell you what region of the length of each of the fragments so that you can calculate going from this end of the DNA to the other end, for example, determine exactly where your protein binds. This is, if you understood the principle of this technique, that's good. You can test your knowledge using the study question at the end of this lecture to ask yourself, can I actually calculate exactly where the protein is binding? Okay? So go home and practice this on your own. Any questions about this? Does that make sense? All right. All right, so the next assay that we're going to talk about, so let me just go back here for a second, because I think we have time to cover this. So, for example, here, how do I know that the TBP is binding between minus 40 and minus 10? Right? Not minus 40. Is binding between which coordinates here is the TBP binding? You can calculate that by looking at the ladder. So how do you do that? So you know that the DNA fragments that are running here is about 40 base pair, right? So that's your ladder telling you. And the DNA fragments that are, so this is not the base pair from TSS, sorry. That's not the ladder. So what these coordinates are telling you is they're telling you the distance from the TSS. So what they're telling you is that, I don't have any space, so let me go like this. What they're telling you is that this is the TSS in the DNA fragment that you used. Here's your TBP binds. This is your 1, right? And then this is your going to be minus 50 here, and then plus 50 here. So the marker that's telling you is that where in this fragment is the length of this fragment. So here would be a short one, and here would be a long one going from minus 50 to plus 50. This will be going from minus 50 to minus 30, for example. Okay? So that's what it is telling you. And then what you are going to do is you're going to look from the distance where the label is produced, label is put, right? And now you're going to look at the length of the DNA fragment. Let's say this is 30 base pair, right? And where is this going to run? It's going to run over here, right? Because it's a short DNA fragment. So the cut here, this fragment is going to be 70 base pair, right? And then it's going to be run somewhere up here, right? Does that make sense? So you can calculate where it's going to run in the gel. And then you look and you say, okay, in the minus 30 to minus 10, I see my footprint of my TBP. So here is the coordinates. You say, here's minus 30 and here's minus 10. And then I determined the binding site of the TBP there. Okay? So hopefully that will help you with this study question. All right. So the next assay, electrophoretic mobility shift assay. So this is what we call gel shift assay. And what the question it's going to ask is, does my transcription factor bind to a DNA sequence specifically? So you know that your transcription factor MyoD, for example, binds to a short sequence, let's say ATGC, and then you ask, does it actually bind to this sequence specifically? Does it specifically recognize the sequence, or can it bind to anything? All right. So for this, you do gel shifts. So how do you do this? The first thing that you do is that you make a DNA probe and you label it radioactively, okay? In this case, your DNA probe is going to be double-stranded, and it's going to be short. In gel shift assays, we make probes about 30 to 50 base pairs or so long, okay? So you make a DNA probe and you label it, and you label it at the end, just like the DNase assay, or you label it inside the DNA by incorporating radioactive nucleotides while you're doing a PCR or while you're synthesizing this DNA, okay? So you incorporate radioactivity into the DNA that you're assaying. And then what you do is you put your protein, your transcription factor, into the tube along with your DNA. And if it binds to this DNA stably, it will produce a complex of DNA transcription factor, right? So you're going to have a TF plus DNA. And you're going to have some free DNA that's not bound, and you're going to have some TFs that's not bound, right? Some transcription factors that's not bound. It's a simple chemical reaction, right? It's binding. You have some free TF, some free DNA, and then some stable complex. All right. So what you do next is you run this DNA and protein in the tube in a gel. And then you detect where the DNA is using radioactivity. Okay? Again, you're detecting everything by radioactivity. So if you take your tube and only put your radioactive probe, and then you run this on a gel, and this is a special gel that doesn't destroy DNA-protein interactions. This is what we call a native gel. Okay? So DNA and protein, when it's complex, it's going to run as a complex in this gel. So if you have only your DNA fragment, what you're going to have is you're going to have your probe running down here, right? So this is your labeled probe. Okay, if you put a protein that you know does not bind to this DNA, right? You put a protein like tubulin or whatever, some protein that doesn't bind to this DNA, and mix it up with this DNA, you also are going to see this band because there is DNA that's unbound DNA in the tube, right? And then your protein is not bound. But when you add your protein in increasing amounts, okay, you're putting more and more protein in each tube, what you're going to have is you're going to have a shift up, because these DNA here are running higher because they are complexed with the transcription factor. Okay? So the transcription factor plus DNA runs a lot slower. So it shifts up that signal. So that's why it's called a gel shift. Right? Make sense so far? Okay. So far, this assay is not testing whether your DNA, your transcription factor is binding to a sequence specifically or not. What it is telling you is that whatever DNA you put in that tube, it's binding to it stably enough to shift, right? So then you need to test, is this thing actually binding specifically? And you can do this in a couple of different ways. One is to use what we call a cold competitor, okay? So what you're doing is that in your tube, you have your labeled DNA, right? And then your transcription factor binding to it. You add a bunch of the same DNA. This is a specific competitor, so that's exactly the same DNA sequence, exactly the same DNA, but it's not labeled with radioactivity, okay? What you're doing is you're increasing a lot of DNA here for this transcription factor will have a chance to come in and bind here, okay? So if you have one labeled DNA and 100 non-labeled DNA, just because of the on and off dynamics of the transcription factor, what that means is transcription factors don't bind and sit DNA for a very, very long time. So what they do is they come off and bind again. Okay, so when they come off from the labeled DNA, the chances of this transcription factor to bind a non-labeled DNA is going to be 100 times higher, right? Because you put 100 times more non-labeled DNA. Does that make sense? Any questions?
When you say the extra DNA that you're adding, are these different sequences?
It's exact same sequences. This is specific competitor. It's exact same DNA, exact same sequences. You're putting it a lot more, like 100 or 1,000-fold more of it, right? But they are not labeled. Any questions?
Means they don't show up.
They won't show up. Excellent, right? So the idea is this. If you put this specific competitor and then the transcription factor moves over, right, and then binds to the non-labeled DNA because there's so much more of it, and then you run this product in a gel, what you're going to see is you're going to see that your labeled DNA is now free, right? So you no longer have that shifted fragment. Does this mean that in your tube there are no transcription factors bound to DNA? Yes or no? No. It's there. You just don't see it because it's bound to the DNA that's not labeled. Okay? You can do the same thing. Now we're going to test for the specificity of the protein, and then what you do is you use a non-specific competitor. A non-specific competitor means you use a piece of DNA that does not have the same sequence. Okay? So you're using a different DNA that does not have the same sequence. If your transcription factor is sequence specific, so it binds only your favorite sequence that you put in the probe here, is it going to go and bind to these non-labeled DNA, non-specific competitor? No, right? So when you take all of these products and then run it on a gel, what you're going to have is you're going to still have your gel shifted band, right? So your protein is preferentially binding to your labeled DNA and not that unrelated, non-specific competitor DNA. Any questions? Make sense? All right. The second way you can test the specificity of your gel shift is you can prepare a probe, right, that contains the sequence that contains the DNA, you make a DNA probe that contains some mutation in your sequence. So if you suspect your transcription factor is binding and you're testing, is it binding to TAA, TCC sequence, right? You can take that DNA and now you can mutate that sequence to TGT, GCC. If the sequence is important for the binding, so you should no longer see any shift if you use that probe, right? And now you don't see any shift here, suggesting that sequence is important. All right, any questions about that?
How do you find the other sequence? Is it just any random sequence?
We typically just randomly use. For example, you just use a little bit like personal judgment. You say, okay, if my protein is binding a lot of A's, right? Why don't we just change it to T or G, which is completely different, right? And it should then interfere with this protein's binding. Good. So we do test for sequence specificity of our proteins because there are some proteins that just love DNA. They'll bind to anything. Right? They don't care, sequence or not. Give me an example. Doesn't have to be a transcription factor. Something that loves DNA. DNA polymerase loves DNA. What else?
Anything with a lysine zipper?
Leucine zippers, those are typically sequence specific.
Oh, okay.
Transposase do love DNA, that's right. What else?
Histones.
Histones, oh my god, right? Those are the worst because they cover 70% of eukaryotic genomes. That means they're not that sequence specific. They're going to go for any DNA around, right? Okay. So the last thing that we're going to learn today is just a glimpse into how these basic techniques have been evolving to actually ask questions genome-wide. So we're going to learn a technique in which you can map all the binding sites for a given transcription factors in the whole genome. Okay? So a lot of these genome-wide techniques, they're based on the principles that we learned in DNA's footprinting assay, in gel shift assay, etc., etc. And here is one that we're going to talk about, which is called ChIP-seq. That's called chromatin immunoprecipitation followed by sequencing. All right. So what we want to know here is we want to know where my protein binds in the genome. So what we do is, for example, here's my protein of interest that I depicted in triangle. And then you have lots of nucleosomes, right? And histone proteins are depicted in these little circles. And there's some other proteins around, but I didn't draw them because it's going to be too much, okay? So you take a whole bunch of cells, and then what you do is your protein is binding at some loci, some places in the cells, very strongly, right? And then in some places it doesn't bind very strongly. So in some cells you find the protein binding to these sites, and in others it's not bound to these sites. So the first thing that you do is you take each of your cells and then you cross-link the DNA to protein. And you can use a small cross-linker called formaldehyde that is going to do this. So those are little X shapes here, right? So it basically stable chemically cross-links your protein to the DNA. All right. So you have cross-linked chromatin. So I'm now showing you only just one chromatin here instead of a whole population of cells. And then the next thing that you do is you do shear chromatin bisonication. What that means is you use sound waves to literally break the chromatin into smaller pieces, right? The human chromosomes are megabases long, right? And you want to make about 500 base pair or pieces. And then you can use sound to do that, to shear everything. And then you separate your sample into two. In one, we're going to call it the immunoprecipitation sample, and in the other, we can call it reference sample, input sample, or control sample. Okay? I'm going to call this input sample for now. All right. So what you do is next, you make an antibody that is specific to your protein of interest. So this antibody is going to only bind to your protein. Okay, so then you use these antibodies to pull out, you separate all the DNA that is cross-linked to your protein and wash away all the DNA that's not bound by your protein, right? So the immunoprecipitation is going to isolate the DNA fragments that contain your protein and not the other ones. Your control sample is going to contain all of the DNA fragments, regardless of bound or unbound by your protein. All right, so what do you do next? Next, what you can do is you can reverse the crosslinks. Formaldehyde crosslinks can be reversed by heat. So you reverse the crosslinks and you isolate, you recover the DNA from each of the reaction. And these DNA are going to be the DNA that were bound by your protein. And this DNA is going to be DNA that's, regardless of binding, going to be isolated. And then what do you do next? You take your chip DNA and then you take your input DNA, and for each of them you create an Illumina sequencing library. You guys learned about Illumina sequencing, right?
In lab, but not yet.
In lab?
Yeah, but not here.
Not here? You should learn. You should have, yeah. Professor Siegel covered that. Did he cover library generation or just the...
Yeah, we did some cDNA, like the gen.
Okay, good. Okay, so I'm pretty sure he was going to do that. So I'm going to summarize this. What you're doing is you're going to prepare your DNA for sequencing for Illumina. Okay, and you're going to do that separately for your chip sample and input sample. So what you do is this DNA is sheared into smaller pieces, so they're about 200 to 500 base pair long. The first thing that you need to do is you need to amplify this DNA. But you can't really simply do PCR because you actually don't know what these sequences are, right? That's what you're trying to figure out. So the first thing that in Illumina sequencing library preparation is that you add an A to the ends of your DNA, and then you take sequence adapters. We call them adapters. They're basically short, double-stranded DNA fragments that contain a sequence that you know, and this sequence is required for Illumina sequencing. And these adapters have a T at the end of a T extension, right? So there is an extra T. So this is not a blunt end. And then you mix these things up together with a ligase. And what it's going to do is that this T and A are going to base pair, and you're going to ligate these adapters to the end of the DNA fragment. Okay? It's kind of like cloning, right? You have sticky ends. By adding an A to your DNA fragment and having an adapter with a T, you created a one base pair sticky end, right? And you ligate it. So here's a question for some of you biochemistry enthusiasts. What may happen, for example, if you don't add an A tail and then try to ligate these adapters, and adapters don't have this T overhang, right? So you don't have these sticky, right? You have two blunt ends. You have a DNA that is blunt-ended, and then you have your adapters that are blunt-ended. Will these things ligate? Can these things ligate? Yes, blunt ends can ligate, right? That's non-homologous end joining. You have two blunt ends, you can ligate. Ligase will work, it's fine. But this is what the beauty of molecular biology is. You have to think the type of products that you want to create. You want to create this product, and you want to increase the chances of creating that product. If you had a blunt end, what could happen is there could be several things that can happen. Your little DNA fragment can ligate to each other, right? Because they're blunt. They can go either way, right? Or your adapters can ligate to each other, right? So you're going to have a whole bunch of undesired products from your reactions. So clever molecular biologists said that if I just simply mix these adapters with my DNA, I'm going to get a whole bunch of things that I don't want. So what I should do is I should make this ligation specific by creating a sticky end, right? Is that awesome? Yeah. Yay. All right, so the next thing that you do is you PCR amplify this DNA. And now you can PCR amplify because you know the sequences of these adapters. So you can use primers that are going to anneal to these ends and then create what we call your sequencing library. So this is going to contain a whole bunch of different sequences that are coming from your chip, an input that are amplified, and they're going to have these adapter sequences. All right. Then you go through Illumina sequencing, and the Illumina sequencing is going to give you short reads. What it is going to give you is that it's going to give you the sequence of the end of your DNA fragment. So what that means is, like, let's say your DNA fragment is A, T, G, C, blah, 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 T, 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 etc. And then you have your adapters here, and the Illumina sequencing is going to give you this sequence, okay? And that's going to be typically short, 50 base pair to 75 base pair, etc. And then you can read from each end of the DNA fragment, so it can give you a sequencing read for this end and the other end as well. So you're going to, when I say read, it's basically these sequences that the Illumina sequences tell you the DNA fragment that you cloned, the DNA fragment that you amplified with these adapters contain these sequences. So what do we do with them? We take all these sequences, which are going to be millions and millions of them, and we're going to ask, where does this sequence come in the genome? Right? You're going to take that sequence, ATGC, blah, 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 50 base pair long, and you're going to say, where do I see that sequence? So this is what we call aligning. So we're going to align those reads to the genome. And the way that you're going to do is you're going to say, okay, in my fragment, I see this sequence at at one end of the fragment, and I see this sequence at the other end of the fragment, and they align to this site in the genome. So this is like coordinates from 0 to blah, 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 right, for each chromosome. And you're going to align your sequence. And you know that your DNA fragment is actually connects these two sequences because that's you're reading the two ends of the fragment, but in fact what you did is you have the fragment that's longer, right? And then you did for this for millions of sequences, so you put all your align all your reads, etc. And then what you do is you calculate a chromatin immunoprecipitation chip binding score. And you do this by calculating a coverage. What I mean by coverage is this. For each site in the genome, for each base in the genome, how many different DNA fragments overlapped with it? Okay? So what's the coverage for this site in the genome, shown here with dashes? 1, 2, 3, 4, 5. What is the coverage? How many fragments, how many aligned fragments are coinciding with this specific base pair here? 2, right? What about here? What about here? What about here? What about here? 4. So what you can do is then you can plot that coverage in the y-axis, right? And the regions that you have many, many fragments are going to appear like a little peak, right? So what are these little peaks? What does this tell you? It tells you that your protein was over here, right? You had your protein over here, so you IP'd all these fragments that contains your protein, right? And that's why it has a high coverage, because you're recovering those fragments a lot more than other regions where you didn't have your protein. All right. So that's basically the coverage is a measure of the binding. So here's yeah?
Why does it not matter that you don't know the sequence in between? Or do you just get that sequence based off of the alignment?
Yeah, you get that. Excellent question. You don't know, but you know the genome sequence. What that means is that, okay, if this was A, T, G, blah, blah, in the genome, I know that it continues as A, etc., etc. So if I'm capturing this and this, I know that my DNA fragment also had this, right?
For this to work, you need to have the entire genome
Excellent.
sequence to fill in the gaps.
Very good, very good. For this ChIP-seq to work, you have to know the genome sequence of the organism that you work with, because you're going to use that genome sequence. Questions?
What exactly is the x-axis?
X-axis is the coordinate, right? So what you have here is for example, this is chromosome 1. It goes from 1, 2, 3, 4, 5, 6, etc., etc., to millions, right? And you're going to have your genes here, etc., right? Make sense? Okay, so here's an experimental result from my lab. So we study a group of proteins called the dosage compensation complex in C. elegans, and we study how these proteins bind to the genome, okay? So, for example, here I'm showing you our ChIP-seq results using antibodies against this protein called STC1. And what you can see here is that when you plot the coverage across the genome, here we're looking at a zoomed out view that's about 100 kb or so. And each of these little blue lines are each of these genes, right? So in the C. elegans genome, I'm seeing where the genes are. And then along the coordinates of this chromosome, I'm seeing that there is one site in this chromosome that is immunoprecipitated by STC1 antibody. So I interpret it as that the STC1 is binding to this site, right? And that's in 3 biological replicates that we have done. You always see this binding site that's bound. And then in the input, which is the control, you're not going to see this fragment pulled out as much more than the others, right? You're not going to see that enrichment because you're basically sequencing all of the little fragments across the genome. So this pattern, the coverage pattern, is going to be much flatter compared to your ChIP experiment. Does that make sense? All right. So here's a question for you. Oh, okay. So what can we do with this data, right? With this data, we can find out which DNA sequences are important for directing my protein to the C. elegans genome. And how do I do that? I don't have just one site of binding. I actually have thousands of those sites. And then what I can do is I can take all the sequences under each site, right? So each of these sites contains some sequence. And then use a DNA program. And I ask the DNA program, look for short DNA sequences, which we call them motifs. Short DNA sequence motifs that you find commonly in these sequences, okay? And then what this program finds me is this motif, which is GC, GC, AGGG, right? And then next in my lab, what we did is we went to that site that I showed you. We mutated this motif using CRISPR, and then we said, does it actually bind now? And we find, no, it doesn't, telling me that this sequence is important for the binding of my protein, right? Okay, here's a question. Does the data that I show you, this ChIP-seq data, does it show that STC binds to the motif directly? I said that I have ChIP data, it binds there. I have this motif that is present at that site. If you mutate the motif, it doesn't bind there. Does it mean the protein that I looked at by ChIP binds to this thing directly? No. Because when you do ChIP, you're cross-linking all the proteins to the DNA, and sometimes what you might find is you have your mystery transcription factor binding to this GC, GC, etc., and then your STC1 binding to it, right? And because everybody is cross-linked, you put a formaldehyde, It's going to cross-link protein, 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 DNA, and then you're going to do a antibody pull down here, and you're going to find that STC1 is binding to this side, but it might not be directly binding to DNA. That doesn't mean my data is bad. That means that my protein is really binding to that side in the genome, but it might not be directly contacting the DNA and binding to the sequence. Yes?
So you're recognizing the entire complex?
You're recognizing the entire complex. So next, I can say, I know the STC1 is probably the protein that binds here because it has this beautiful DNA binding domain, and I need to find out if it directly binds or not. What technique can I use to do this? What technique can I use to test if my STC1 protein is directly binding to this sequence or not? Flip, 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 backflip your notes. What did we learn?
Could you mutate the DNA binding domain and see if it's still...
You can mutate, but that doesn't mean that it's STC1 binding. What else can you do?
A mobility shift.
A mobility shift. What else?
You do fish and
Fission immunofluorescence will tell you if they co-localize. Right? It's going to be exactly like the chip experiment. It's going to tell you that they are at the same spot, but it's not going to quite tell you if the protein is binding. So gel shift is one. You can see if it shifts my DNA, then I know it's direct binding. What else? There was another one in which we mixed the DNA with the protein, and we put some enzyme to cut the DNA. It looked pretty complicated, and I said, look at the study question and do it yourself. DNA's footprinting assay, right? If my protein actually protects this sequence in a tube, in vitro, outside the animal, right? In a tube, I put my DNA, I put my protein, it binds. I can use DNA's footprinting to see if it's binding or not. All right, here's another example. So here's, again, our chip data, and what we're looking at different proteins binding, and then we're looking at RNA polymerase II binding, and then this is our negative control, and here are our genes. Which of these genes is being transcribed? Name one. BET2, right? And how can I tell that? Because I know that RNA polymerase II is right on that gene transcribing it. Because I chipped RNA polymerase II and looked where it binds, and it's binding to this gene. Where do my condensins like to bind in the genome? What kind of gene regulatory sequences does it like to bind? Come on, come on. What you can see here is that here's the BET2 gene. This is the direction that it's being transcribed because I see these arrows. And right upstream of the gene is the promoter, right? And that's where I'm seeing my binding peak. So it looks like my proteins like to bind the promoters, right? And because that pattern looks so much different than from RNA polymerase, right? RNA polymerase is binding from transcription start side to the end side, but my protein likes to bind at the 5' end of the genes. It's the beginning of the genes. All right, that's it, and I see you probably Friday. Good luck, study, and you'll do well, hopefully.